In 2001, archaeologists excavating a medieval cemetery in Central Europe pulled a fragment of bone from the soil. It was small, unremarkable, the kind of thing that gets cataloged and set aside. But when the ancient DNA inside it was finally sequenced decades later, one result stopped the researchers cold. The individual had carried type AB blood. That alone was not surprising. What was surprising was everything else the DNA said about who that person was, where they had come from, and what their existence meant for a blood type science had always treated as a simple random accident of genetics. For decades, the explanation for AB blood was settled and clean. It was a mutation, a fluke. The youngest of the four main blood types appearing only about 1,000 years ago when populations with A and B blood happened to mix. The story was tidy. It fit the textbooks. It was also the ancient DNA record is now making clear, far too simple. The real story of AB blood runs deeper than a random collision between two gene pools. It tracks human migration, survival pressure, immune adaptation, and the slow rewriting of the human body across centuries of contact and change. What the bones are now telling us is not the story we were given. It is stranger, older in its implications, and more revealing about who we are than anyone expected. There are four main blood types. Most people learn this early and never think about it again: A, B, AB, and O. The differences between them come down to something almost invisibly small: proteins on the surface of red blood cells called antigens. Type A blood carries A antigens. Type B carries B antigens. Type O carries neither. Type AB carries both. Those antigens are not decorative. They are identity markers. The immune system reads them constantly, checking every cell that passes through the bloodstream against a mental map of what belongs and what does not. When a foreign antigen appears, the immune system raises an alarm and begins producing antibodies to destroy the intruder. This is why blood type matching matters in transfusions. Give a type A patient type B blood, and their immune system will attack it. The consequences can be fatal. AB sits outside this logic in a way that has fascinated immunologists for generations. Because AB blood carries both antigens, the immune system of an AB individual never learns to treat either one as foreign. It recognizes everything. It attacks nothing. That makes AB the universal recipient for red blood cells. In an emergency, an AB patient can receive blood from any donor on Earth, regardless of type. But here is where the paradox begins. That same tolerance, that biological openness that makes AB so valuable on the receiving end, is the product of a genetic combination so rare that for most of human history, it simply did not exist. Type O is ancient. Geneticists believe the O allele has existed in the human population for hundreds of thousands of years, possibly longer. Type A and Type B are younger but still old, emerging hundreds of thousands of years ago as separate mutations in separate populations on separate continents. They persisted, spread, and became fixed in the gene pool across timescales that dwarf recorded history. AB did not exist until roughly 1,000 to 1,200 years ago. In evolutionary terms, that is not recent. It is practically this morning. The mechanism that produced it was not a mutation in the traditional sense. No single individual was born with a spontaneous genetic change that created AB from nothing. Instead, AB appeared when something that had never happened before at scale began happening everywhere at once. People who carried type A blood and people who carried type B blood began having children together in large numbers. The offspring of those unions could inherit both antigens. When they did, AB entered the world, which means AB blood is not a random accident. It is a document. Every person alive today carrying AB blood is carrying genetic evidence that at some point in the last 1,000 years, their ancestry crossed a boundary. Two populations that had been separate long enough to develop different antigens came together. The child that resulted carried both. For centuries, no one had the tools to read that document properly. Ancient DNA has changed that. And what it is revealing about where AB came from and why it appeared when it did is rewriting the map of human movement entirely. The leading theory for how AB blood spread across the human population points to one of the most dramatic episodes of enforced human mixing in recorded history: the Mongol Empire. Beginning in the early 13th century, the armies of Genghis Khan swept west and south out of the Central Asian steppe, eventually controlling a territory stretching from the Pacific coast of China to the edges of Eastern Europe. It was the largest contiguous land empire in human history, and it moved populations on a scale that had never been seen before. Populations carrying predominantly type A blood in Europe and populations carrying predominantly type B blood across Central and East Asia were suddenly, violently, repeatedly brought into contact. Soldiers, slaves, merchants, and survivors intermarried across boundaries that had been closed for thousands of years. Children were born carrying genetic material from both sides of those boundaries. Some of them carried AB. Ancient DNA recovered from burial sites along the old Mongol trade and conquest routes is beginning to confirm what the theory predicted. AB frequency increases in the archaeological record in precisely the regions and precisely the centuries that correspond to peak Mongol expansion and the trade networks that followed it. The signal is not uniform, it is not clean, but it is there. What the DNA is also revealing is something the Mongol theory alone could not explain. AB did not just appear in one place and diffuse outward. It appeared in multiple locations across a relatively narrow window of time. That pattern does not look like diffusion from a single source. It looks like convergence. Multiple populations, multiple crossings, the same result written into the blood independently and simultaneously. The ancient world was moving faster and mixing more completely than anyone had mapped. The immune system of a person with a B blood is, by any conventional measure, unusually tolerant. It does not produce antibodies against type A antigens. It does not produce antibodies against type B antigens. It has been trained, by the simple fact of carrying both, to leave both alone. That tolerance is what makes AB the universal recipient. But immunologists studying AB blood have spent decades asking a harder question: what else does that tolerance do? The answer is complicated in ways that are still being untangled. Because the immune system is not a single switch. It is an interlocking network of responses, each one calibrated against what the body has learned to recognize as self. When that calibration is set wider than usual, as it appears to be in AB individuals, the effects ripple outward into systems that have nothing obvious to do with blood transfusion compatibility. B individuals show measurably lower rates of certain autoimmune responses. Their immune systems are less likely to misidentify the body's own tissue as foreign and attack it. That sounds straightforwardly beneficial. In some contexts it is, but immunological tolerance cuts in both directions. A system less likely to attack the wrong target is also, in some configurations, less aggressive toward the right one. This is the paradox at the center of AB blood. The same biological architecture that makes AB individuals universally compatible, that made them potentially more survivable in a world of mass migration and new pathogens, also appears to carry trade-offs that researchers are only now beginning to measure. The tolerance that opened every door also left some doors it perhaps should have closed. The biological footprint of AB blood extends well beyond the immune system, and the further researchers look, the more unusual the picture becomes. Start with clotting. People with AB blood carry higher levels of a protein called von Willebrand factor, a clotting agent that helps seal wounds by triggering the cascade that forms a blood clot. Higher levels mean faster clotting. In the immediate term, that is protective. A cut stops bleeding more quickly. A surgical wound closes more efficiently, but the same mechanism that accelerates clotting at a wound site can, under certain conditions, accelerate it elsewhere. Research has consistently found that people with non-O blood types, and AB in particular, carry a measurably elevated risk of deep vein thrombosis and pulmonary embolism. blood clots forming not at wounds but inside veins and arteries where they have no business forming. Then there is the brain. A 2014 study published in the journal Neurology followed thousands of adults over an extended period and found that AB blood type was associated with a significantly higher risk of cognitive decline in later life. The researchers proposed that the same clotting dynamics affecting the cardiovascular system may be affecting blood flow in the brain across decades, quietly and cumulatively, in ways that only become visible in old age. The gut adds another layer. B individuals tend to carry a microbial profile that blends characteristics found in both A and B types. That mixed microbiome appears to influence digestion, metabolic efficiency, and immune response to infection in ways that researchers are still mapping. The body of a person with AB blood is not simply rare, it runs by a different set of calibrations entirely. One thing before the final piece, everything I just showed you about AB blood is traceable inside your own DNA results. I wrote the guide for exactly that. It's called Where Do You Really Come From? Link is below. Now, here's what nobody talks about. The science of AB blood is no longer just a curiosity sitting at the edge of immunology. It has moved to the center of one of the most consequential shifts in modern medicine, the move away from treating all patients the same and toward treating each patient according to the specific biological architecture they actually carry. Precision medicine, as the field is now called, uses genetic and biological markers, including blood type, to predict how individual patients will respond to treatments, medications, and surgical procedures. AB blood has become one of the more studied markers in this emerging framework, precisely because it behaves so differently from the other types. Some chemotherapy protocols show measurably different efficacy profiles in AB patients. Certain vaccines appear to generate stronger or weaker immune responses depending on blood type. The data is early, but the direction is consistent. Blood type is not just a transfusion variable, it is a window into how the whole system works. Stem cell research has added another dimension entirely. Because AB stem cell lines provoke fewer immune rejection responses, they are among the most versatile raw material available for developing universal therapies. Laboratories working on lab-grown tissue and organ replacement have found AB donor material disproportionately useful precisely because of the tolerance paradox that makes AB blood so immunologically unusual. In the early months of 2020, as laboratories around the world raced to understand why some people were being devastated by the new COVID-19 while others moved through it with barely a symptom, a signal emerged from the data that nobody had specifically gone looking for. Blood type appeared to matter. Multiple independent studies across different countries and different patient populations found the same pattern. People with type O blood showed a modestly but consistently lower risk of severe illness. People with type A and type AB showed a modestly but consistently higher risk. The effect was not large enough to be determinative. Having AB blood did not sentence anyone to a severe outcome, but the signal was real, it was reproducible, and it pointed at something researchers are still working to fully explain. The leading hypothesis connects back to the clotting dynamics that AB blood is already known to carry. The most dangerous presentations of severe COVID-19 involved not just respiratory failure, but widespread clotting events throughout the body. A blood type already predisposed toward elevated clotting activity may have had less margin when that system was pushed into overdrive by infection. The pandemic finding did not change what AB blood is, but it added a new chapter to the case that blood type is a variable medicine cannot afford to keep treating as administrative background noise. Meanwhile, in laboratories in the United States, the United Kingdom, and Japan, researchers are making steady progress toward synthetic blood. Engineered to be universally compatible, carrying no antigens that could trigger immune rejection, synthetic blood would effectively make every patient a universal recipient. For rare types like AB, the implications are significant. The scarcity that has always defined AB donation, the small pool of compatible donors, the logistical pressure in emergencies, could eventually dissolve entirely. In 2001, a fragment of bone came out of the ground in Central Europe. Inside it, a DNA sequence that nobody expected, a blood type that, by the timeline science had always accepted, had barely existed long enough to leave a mark on the archaeological record. And yet, there it was, written into a medieval skeleton with the same permanence as everything else the body remembers. That is what AB blood ultimately is. Not a fluke, not a footnote, a record of movement. Every person carrying AB today is carrying evidence that somewhere in their ancestry, two populations that had been separate long enough to diverge found each other. A boundary crossed, a child born on the other side of it. The blood remembers the crossing even when the history books do not. The science is still moving. The ancient DNA record is still being built, cemetery by cemetery, fragment by fragment. The connections between AB blood and cognition, clotting, immunity, and disease risk are still being mapped. The synthetic blood that could render the rarity of AB irrelevant is still being engineered. Every answer the research produces opens three more questions. But the oldest question, the one that textbooks thought they had settled, has already been answered. AB blood was never a random accident. It was the immune system's response to a world that was changing faster than any single population could track alone. The bones knew. The DNA confirmed it. We just needed the tools to read what had been written there.